Walk with me through the cellar door.
A storm is coming, Francis.
A portal to a more skeptical world.
Cellar Door Skeptics begins right now. Prepare for the revolution with your hosts, Christopher Tanner and Chris Hanna.
Welcome to another Cellar Door Skeptics. Thank you all so much for joining us tonight. We are doing a very special episode tonight. And I guess it's not really night because you can see the light in the background. So it's technically not light or night in this world here. But I thought it would be fun because we promised everybody that we wanted to do a vaccine episode. And we promised we had an expert that was going to come on and have a conversation with us. And we didn't get to it last recording. So I thought we'd do a special recording where we'd be able to allow for her to come and join us today. So thank you so much for joining us, Doctor Abbie. How are you tonight, ma'am?
So tired. How are you guys doing?
Pretty good. I don't have the same rigorous schedule that you have. Apparently. So, my late night habits just got worse because technically I don't have to get up at 6:30 in the morning anymore.
Oh no. COVID has altered everyone's normal routine, and mine has shifted just a bit earlier than normal, and it's gotten a bit more hectic. But hopefully I can share some of the information of the stuff we've been doing with your all's viewers.
Awesome. And for those of you who don't know, so Abbie has been on the show, I believe, 3 times now in the history of our show, both shows that we used to have. And essentially, she is a research scientist at Emory University who normally will research HIV, but currently, given the crisis, has been helping out with the COVID vaccine, I guess, right?
Kind of. So yes. So normally I study the virology and immunology of HIV and try to figure out how to stop new HIV infections. But every single lab in the country is shut down right now unless they are performing COVID research, essential research in relation to the current crises that we're currently experiencing. So right now, I work with a group of physicians at the Emory Hope Clinic, and they facilitate all these really cool trials that you guys, I'm sure, have been hearing about in the news, all of these COVID drug trials, COVID vaccine trials. These are my friends and collaborators who are running these. And so what some of you all might not understand is that I am a research scientist. I'm a PhD research scientist. And what we do is very much sort of the Wild West. If we get a weird idea or something cool runs through our head, we can go, hey, yeah, let's try this. Let's do that. And we can do that. But clinical research is very, very different. So, for example, let's say something as really a very straightforward protocol in the lab of, let's say, purifying cells from blood. That's something I have done, for decades at this point, just bags and bags and bags of blood I've purified cells from. All of that experience doesn't really matter in the context of clinical research. So for me to help with clinical research, even with all of my experience, I have to do all of this testing, go fill out all of these forms, jump through all of these hoops. So I am helping doing little things like that with the clinical research. But like these are massive big trials that are going on. And I am literally just helping like the shake and bake commercials. And I helped. That is what I'm doing. So I do not want to give the impression in any way, shape, or form that I am a ringleader of any kind in all of this. No, these are massive trials with lots of people contributing, and I am helping a tiny bit.
Yeah, and I think that's kind of the important thing here, right? It's not all about one person. It's about a bunch of
No.
people.
Yes.
Wait, what?
Yeah, well, we know, buddy, you think it's always about you, but it's
I
not.
mean, I wasn't gonna go down that road, but all right.
Well, I would say this, in some there is the idea of, kind of the rogue scientist, or the wild and crazy scientist who's got this one crazy idea. It's not really the way modern biological sciences, biomedical sciences work. And I think, you can go back and look at what was that blood testing company, Thanos or whatever, with Elizabeth Holmes, who is trying to say that, oh, she was this rogue who was going to upset the industry. That's not really the way biomedical science works. The way it works is lots of people working together towards the same goal.
Yeah, exactly. And so for those of you who want, we've thrown a link in the description as well of the university that she works for. And they have some information in there as well. We're going to go through some clinical trial information. And I think that's part of one of the big reasons we wanted to have you on, because there's a lot of fake news out there, right? And, the last episode, one of the things that we went through, and Buddy can attest to this, right, is that we went through some tweets, where people are thinking that the fake news, or that the cell phone towers are causing this, or why are all of a sudden 5G towers popping up around the world when we should be quarantining? And it's like, well, whether that's safe or not is inconsequential, but that's not what's causing the freaking virus. Like, come on, people. This is not a Stephen King novel.
Well, yeah. The conspiracy theories running amok because there's so many internal biases and people just not wanting to accept the fact that maybe they're wrong.
Well,
Yeah.
and viruses and vaccines have always been sort of like incredibly attractive to conspiracy theory minded individuals, and so this is something that they have fed on.
Hmm. Well, yeah. I mean, you look at any of the anti-vax rhetoric, and it's just any sort of little wiggle room. They'll just try to get their finger in there and just make it as big as they can, just to try to draw as much attention to that one small insignificant detail. But when it's found out that their I don't know, demagogue has been but lost his license because he was there was conflicts of interest. Oh, no, that wasn't it. That's a cover up, blah blah blah. It's so everything, it has to fit into their worldview, even if they has to smash it with a hammer to make it in there, and they lose a good chunk of the story.
Exactly. And that's, that's exactly why we wanted to have an expert on, because we've talked, for the past month, we've talked about different things, that people are trying to do, talking through, the discussion of what's really out there and what's really going to help maybe put an end to this. Because I think the big thing that people are scared of, right, is the fact that this continues to happen. This continues to go on, and their fearless leader keeps telling them it'll be over, and now he has to keep recanting his statements. And this week we found out that Boris Johnson, who everybody thought was fake news, him getting it, he's just faking this, got admitted to the hospital, right?
Well,
so
and
they
now he's
officially
in the ICU.
Oh, now he's in the ICU? Okay.
Literally, while we were prepping for this show, it was released that they transferred him to the ICU. So it's incredibly serious.
Yeah. So that the thing of these people sitting here saying, "Oh, it's just this, it's this conspiracy theory of cell phone towers," or we're gonna have to start taking all these weird drugs to fix it. It's wrong. We got to stop with that and actually bring, what does the science say, and where can we understand and consume this information. So welcome to the show, Abby. Now you know why you're here.
Yeah. So let's start at the very beginning. So 4.2 billion years ago. No,
let's not go that far back.
So I would go back a little further and say something like this. Just this kind of pandemic has been predicted by scientists and epidemiologists for a very long time. At this point, is an inevitability due to humans encroaching on more and more habitats, global warming, animals coming more into contact with humans, and so forth. So individuals have been asking me for layman's resources for viruses and pandemics and that sort of thing. And a book that I haven't seen many people know about or recommend. So a little someone who's named Peter Doherty, and not the one you're thinking of who is in a band, but Peter Doherty is a virologist, a Nobel Prize winner. He wrote a book called Pandemics, What Everyone Needs to Know. And I will send you a link to the Amazon so you can make sure you all are finding the right ones. But he wrote an excellent layman's book that explains some of the basics of science. And he has chapters specifically on SARS and MERS in here. And this was written, let me see, I want to say 2013-ish, I guess it was published. It was published some time ago. But we knew something like this was going to happen. So this isn't like, oh, this is biowarfare. Like we didn't, this has to be genetically engineered because something like this could have never happened. No, we knew something like this was going to happen. It was just, many places were not appropriately prepared and did not necessarily take it as seriously as it could have been. And frankly, at the beginning, I was one of these people as well. I mean, in early March, we were supposed to have a big conference in virology in Boston. I was ready to go. They canceled at the last minute, and I was so disappointed. In retrospect, I am so glad that they canceled that conference. But something like this was bound to happen. But we just didn't know what it was. So when you don't necessarily, something's coming, but you don't know what, it makes it very difficult to prepare. So the question, well, why didn't we have a vaccine already ready? Well, viruses are so diverse genetically, you can't have really like something ready to go and already have the safety data and everything before you know what you need to protect against. And really the same for drugs. Viruses have sort of a playbook. So you have DNA viruses, you have RNA viruses, you have viruses like HIV that carry their genome as RNA and then turn it into DNA. And so we have a number of drugs already that know how to attack that particular step in the virus's life cycle. And these work particularly well, I guess you'd say, for viruses like HIV that go from RNA to DNA, or in the case of COVID, COVID has an RNA genome. Well, humans, we don't have RNA as like a storage mechanism that's a message. And so in theory, if we've got a medication that can interfere with that step in the virus's life cycle, we can really target the virus and have limited side effects to the human. And so one of the drugs that we are currently testing that I am going to pronounce, I'm going to put the emphasis on the wrong syllable. Let's see. Hold on. Remdesivir. That is one of the clinical trial .gov links that I sent. So that interferes with the RNA step in the virus's life cycle. But the key is, you have to have enough of that drug in an infected cell to make sure that the virus incorporates that instead of what it wants to incorporate. You have to make sure it's in the right cell, in the right tissue, at the right concentration, and you have to make sure it's in the human at the right time, because if it's too late, then it's not going to do anything. But what's cool is this drug was actually already developed some time ago in response to the other viruses related to COVID, so SARS and MERS. And we actually already have some safety data on it as well, because during the Ebola fiasco several years ago, Again, we were just trying any drug that we had off the shelf to see whether it did anything to help the Ebola patients. And it turns out that this drug didn't do so well against Ebola, but it was relatively safe for the individuals who got it. So now we can try it again for COVID patients. And so this particular trial, and I really wanted you all to be able to see what a drug clinical trial looks like. Because in the news recently, we've had a lot of reports of, oh, well, we gave these 6 patients who are dying, this medication and this happened, or these 4 individuals, we gave them this medication and something happened. It's very difficult to make any useful interpretations about the safety or usefulness of a medication based off these extremely small, and I hesitate to even call them studies. Really, it's more compassionate use is what that's called. So let's say someone is...
Which one, which article do you want me to bring up?
Oh, I'm just talking about the news in general.
Oh, okay.
Like when you hear some of these very small reports that are coming up, what will sometimes happen in these emergency situations is it's called compassionate use of a drug, where someone is literally dying. They are literally dying. And their physicians are literally throwing up their hands and saying, "Welp, we got to try something. Do you consent for us to try something?" And so that's kind of what I would really call them, more reports as opposed to actual studies. With the clinicaltrials.gov that you just had up on the actual trial, so this involves hundreds and hundreds of individuals, and it's going to be at many different sites around the country, and I believe also around the world will be enrolled. And it's going to be double blind, so the patients and the physicians won't know who is getting which therapy, or whether they're getting placebo. So placebo controlled is always very important. And so, oh no, the other one, sorry. The other clinicaltrials.gov. You're showing the vaccine one. There you go. So double blind, placebo controlled, hundreds of participants. And so once they meet certain enrollment goals, I guess you call them, they're going to unblind some of the data. And let's say, for example, that unquestionably this drug is beneficial to the patients. And this is an emergency situation. So then it becomes unethical to continue giving people placebo when you've got an effective therapy. So then you change the study. So it's called an adaptive study. You change it. And okay, so now the drug becomes our standard of care. And then we can change other things. We can give the drug in addition to another drug. We can give the drug earlier. We can start changing things. So we aren't restricted to always having a placebo the entire time. Also, let's say you unblind it at one point and it's doing absolutely nothing. You can put a stop to it. Or worse, let's say the drug is making things worse, which absolutely could happen. You can stop the treatment as well. So this is what a real study looks like. It's not for people. And yeah, double blind, placebo controlled.
So what you're telling me is that this actually has to be something that we can prove to work in more than just, a small handful of cherry-picked trials.
Yes. Well, or these preprint things. These preprint things have gotten very popular in COVID research, and I have no idea what's going on. I think in theory, preprint. So let's say, I submitted an article, a manuscript of our results, and I submitted this for review, for peer review, sometime in early March. And reviewers get several weeks to give their reviews back. And then let's say I get about 6 months or so to respond. Like, the actual publication process takes a very long time. So when things are very urgent, and in this case things are, I understand the concept of these pre-review, pre-print release of data. But I don't think the general public necessarily understands that. And so we've seen the release of some of these unpeer-reviewed preprint anecdotes, really, that I don't think the general public really understands the limitations of, that I get a bit concerned about.
Yeah, and I think that's that's the problem, is because people are getting their information from a 74-year-old person who's never worked in healthcare his whole life.
Who's never worked in his whole life?
Yeah. Well, no, all right. Anyway, but the difference here is that what he should be doing is bringing people like from your group into the White House and say, hey, this is what we're doing. Somebody should be telecasting in to say, hey, this is what we're doing. This is what we're working on. Let me explain to you. Because yeah, we're going in a little more detail, honestly, than a lot of other, places may. But somebody could easily sum that up and talk on the air and be like, here's the video of whoever this is, and come present this information to the public. Because if somebody were to tell, and this is why I think this is extremely important, if somebody were to have that conversation and say, hey, here's all the information you need to know, broadcast it. Like, how many millions of people are watching, the White House every day? Yeah. I don't, but a lot of people are. Yeah. And if it could be
incredibly educational and informative and calming. So, and that's part of what I would say is strong leadership, is saying, I don't necessarily know what's going, or I don't know what to do in this situation, but here's a person who does, and here's what we're going to do, and have that person step up and explain what's happening. There are incredibly knowledgeable individuals in the United States working on this project that are very passionate and I would say able to communicate this knowledge well. There are interviews with people, at my institution. So one of the top people at Emory, his name is Carlos Del Rio. You can find lots of Q&As, Facebook Live Q&As with him, communicating information about this virus to the general public. They're great. And I wish the government in my state and in the United States would take advantage of these resources.
Exactly. And, that's the thing. Without those resources, people just walk blindly, and claim cell phone towers are, causing
people
COVID.
Are scared. People are really scared because I would say, even in my position, I get panicky. Allergies are so bad in Georgia right now. And I started getting a little wheezy this weekend, and I started panicky, even like just with all of my knowledge, like it is a bit frustrating that we don't have stronger leadership that's in control of this situation and easing people's fears and anxieties with knowledge, as opposed to just these very strange platitudes.
Yeah. So I think,
well, that's part of the problem with like such a scientifically illiterate population too, right? Is that people are very unaware that there's a lot of gray zone. There's there's times, there are procedures and everything, and they're put in place for the safety of everyone. But it's just easier to sit there behind a podium and just say, no, everything's great. We're doing great work. Everything's great. It's fine. It's great.
Yeah.
So I think if we could go through this, because I have a couple of questions, and just you'll have to excuse because I don't have a doctoral and I didn't do this.
And I might not know the answer, so I will try.
All right. So when they talk about a polymerase chain reaction, what specifically does that mean?
Polymerase.
So what's that?
Polymerase. Where are you reading that?
So it's line 6. So line 6 says,
okay. Polymerase.
As a laboratory confirmed the SARS infection determined by that specific chain. So I think that's important.
Yes, so there are different ways we can figure out whether you are infected with a virus or whether at some point you were infected with the virus. And those are two very different things. So in this instance, when you are using the polymerase chain reaction, what you're doing in this particular assay is looking for the virus's genome. You're looking for the of the virus's genome. So that's extremely unique to the virus. And like, let's say it mutates a little, you can tell, your virus is different from perhaps someone else's virus or something like that. It's very, very, very finely detailed. And there's no way to confuse that with, let's say, your DNA or something like that. It's very unique and very specific. There are other ways to look for the virus. You can look for the virus's protein. And then so the thing is with that, though, there is a very small window where you can find the actual virus. And even then, you might even get like a false negative. I think a false negative would be more frequent than a false positive with this kind of test. Because if the swab isn't like I'm sure you've seen the videos of how they do this test. If they don't if they don't swab just right or get in the right nook and cranny, they might not scrape enough the mucus or cells to get a signal to see the virus. And that's only when you actually have viral replication happening right then. Let's say it was 2 weeks later and you've already gotten better. Like, how do you know whether you were infected with the virus? Because I mean, the virus is long gone at that point. So in this case, you can look for the immune response to the viruses. So really, like, that would be the ideal test. So let's say you were doing like a quick screening for HIV. You have someone come in the lab and you swab their mouth with a little swabby thing, and you can tell them whether they have called antibodies to HIV. So these proteins that will stick to the outside of HIV. That's super fast, super easy, not intrusive. If we could get a test like that for COVID, that would be so awesome. I would be so happy. And that would be great too, because apparently lots of individuals have been infected with this virus, had very mild symptoms or no symptoms. And so let's say my case right now, allergies are driving me nuts. I'm paranoid that I might be sick. I don't want to infect my coworkers. I'll stay at home from work. Well, maybe I already got COVID, I don't know, in December when I went with my family to Disney World and I didn't even know it. Well, antibodies would tell me that. And then I could confidently go to work knowing that, I'm not going to make anybody else sick and I'm not going to get sick. So they recently, the FDA just last week approved a test to look for those antibodies. It takes 15 minutes for this test, but you can only do one at a time. If you can see a video of Trump playing with one of the machines on YouTube, and the machine is sideways, but that's a different story from a different day. But yes, the polymerase chain, that's a very long way to say the polymerase chain reaction is looking for the virus's genome. And so you will only be positive in this test if you have actively replicating virus at the time they're swabbing. And it's possible to get a false negative on this test, I would say.
So that makes a little bit more sense, in terms of what we, what we're looking at, right? Because I think that's a big thing, because a lot of people are saying, hey, Chris, or hey, whoever, why can't we make more tests available? Well, it doesn't always work like that. And, to examine certain things, it's just not as, it's not as beneficial. And I'll give an example. I've been quarantined, almost completely quarantined for a month now. Mine started out voluntarily at the beginning of March. After the first week of March, when I flew back from Dallas, I voluntarily quarantined and have been home. And I didn't go out of the house after that Saturday for over 2 weeks. Other than one time I went up to the store, or not to the store, to my work to grab some documents I needed for a project I was working on. And in there was 3 people. Other than that, I had been 100% quarantined, right?
Mhm.
So the test would be meaningless, but everybody thinks I got symptoms because I cough because of my medication.
Yeah. Yeah.
And, I have allergies really bad too. And right now
real
is the
bad.
worst time,
Yeah.
So I think that's why I want to go through this, because if people don't understand why you can't just create mass tests,
Yeah.
they don't they
Well,
won't.
the test that is widely available right now is uncomfortable, intrusive, and it, difficult to really do a mass scale with. Ideally, so like again, comparing clinical test to a research level test, there are things I could do in the lab that would be super fast, super easy, super cheap, could do it large scale, but none of that would meet the appropriate threshold for an actual clinical test. When you are actually diagnosing, preventing, treating disease, there is a much higher threshold that needs to be met for these tests. So it's not.
It's like
They
those
are.
P kits, right? Like you could go get a home testing kit for drugs, and it'll tell you if you've done drugs in the last month. And a lot of people use them. They're like 15 or $20 or something like that. But they aren't the same grade as going to an actual lab and having somebody, either take hair samples or urine samples and send them out.
Yes, I'm sure it's a similar thing. Um, so it's not because these testing companies are being difficult, or they just want to charge more money, or yeah, well, no, they are forced to meet a very different threshold for a very different reason. If somebody doesn't get the right treatment because their test told them the wrong thing, then they get in trouble. Whereas, in a research lab, like I said, it's the Wild West. I can do quick and dirty and it's all right.
Yeah. Yep. Oh man. I don't know. So I think that's interesting because, some of the other things that, you pick out of here is the different types of things. If you go scroll back up, or I'm sorry, I can scroll back up. They give different time frames, right? And so I'm assuming that throughout the trial, this clinical trial period, they're testing different things. Is that correct?
Yes.
So we won't have to go through every single one of them because there's a lot here. But the one thing that I kind of called out or wanted to call out too was the adversary of events, right?
Yes, absolutely.
So can you explain adverse events and how that interacts in terms of that type of a trial?
So you have adverse events and you have serious adverse events. And I am not the best person to ask about this. We have excellent administrative people at the Hope Clinic who are absolute experts at this that I can probably put you in contact with. But the idea is, we are looking at things that, let's say, seizures. We are looking at death. We are looking at heart attacks. We are looking at we're looking at minor things like tripping and falling. We are looking at every single strange thing that occurs because, let's say, for example, that again, just as a hypothetical, that maybe, we give individuals this medication, and all of a sudden we're seeing lots of these minor falls, very minor falls. And it's not a major adverse event, nobody died, not a big deal, but maybe that's telling us something like, oh, well, maybe this drug is having some effect on a neuron, is having some effect on the cerebellum, is having some effect on the brain that we didn't anticipate, and it could cause some larger problem down the road. So it's not just the major events that we're worried about. We're also looking at the minor events. And of course, this is once again where placebo is incredibly important because let's say, you have a number of heart attacks in the drug treatment group. are those heart attacks attributable to the medication, or is that attributable to just the disease course? And you have similar events in both the placebo and the drug treatment group.
Okay, and that makes sense, and that's the funny thing with that, right, is and then this kind of leads me to another thing, something that came up recently in a conversation that I saw on Facebook, and I think we talked a little bit about it on the episode. I can't remember. I guess Keith, you can tell me if I'm wrong or not, but I had somebody that essentially people are talking about why they're not using death certificates appropriately, and they're claiming everything is COVID, so they're inflating numbers.
Oh, yeah. So I actually saw one of those on Facebook too. And oh my God, I just had to scream because this person was like, oh yeah, they're testing dead people for COVID antibody, which, okay, they didn't even say like COVID antibodies or, no, they had antibody as 2 words, there's no like, at the time of this post, first of all, there wasn't even an antibody test for COVID. Second of all, like you don't, oh my God, nobody does that. Like, I have no idea where they even got it in their brains that somebody was ever doing anything like this. But according to them, they're testing dead bodies for COVID antibodies. And if the dead person had COVID antibodies, then they were, on their death certificates, they were saying they were COVID.
Well, and that's what the infuriating, that's what bothered me, is because it's like, why would anybody make this up to inflate numbers, right? I mean, that seems silly to me.
So dumb.
But
it seems silly, but that's because you think it through, and you're not somebody who's sitting there trying to say, no, there's an agenda behind it, so your argument supports my argument, even though it doesn't.
They made antibody 2 words. They made it 2 words.
And can you even? Sorry, forgive my ignorance, but can you actually test a dead body for antibodies?
I don't know. I guess. Sure. If it's...
all I know is that I recently had my blood checked for various antibodies because I just going through and want to make sure that all of my vaccines are up to date and all of that. And I found out that I still have to get 2 topped up or redone or whatever. But I didn't really fully understand the way that it operated. Like if it had the antibodies showed up as a response, or if the antibodies were just always there. That was just my curiosity.
I've got to say, like, I've got to preempt this by saying I've never performed an antibody test on a dead person or with a dead person. So I'm not sure, like, if given a dead body, I could figure out how to look for antibodies in it, I'm sure. But it wouldn't necessarily be my front line. I would prefer a live person,
Right.
just because it's easier. It's easier. You just take some blood or even just spit. So I guess if they were still wet, if you had a good, if your dead person was still nice and moist, you could probably.
and I can feel like half of our listeners just cringing, going, "Oh." If they're a soggy
But
mattress,
eventually,
it
like
works.
the proteases and stuff would start tearing. Yeah. So you have like enzymes that'll start chewing up the protein. So I'm not sure how long. Yeah.
I mean, you would think they would break down to some extent, and they would become something that you couldn't use anymore.
Yeah, because antibodies are very structurally dependent. So you need the antibody to maintain its structural integrity to work well.
Mm-hmm.
So now this is all I can think about now. How am I
going? Do I need to get a picture of a body on the screen for you? Maybe that'll jar it.
Let's see. If it was a freshly dead body, I think we could do it.
just dead sounds. Okay. Anyway, so we're gonna move on from that because I don't think we're gonna ever find a dead body you can test that theory on. And I don't know if you would even want to if you could, right?
No, I don't think it meets the current priority COVID research threshold.
Yeah. I think one of the things too, and this is, I guess this is a little off topic, but one of the big things, in my opinion, that we have to look at too is like what, because, and again, this does deal with dead bodies too. What happens after you die? How long does the virus stay attached to you, right? Because if we think
Um,
about it, there's all sorts of discussions, and that's how other viruses have spread before, right?
Well, to my understanding, I mean, major caveats here uh, is that COVID is not primarily a bloodborne pathogen. So it's not, for example, HIV, which you would find, for example, circulating within the blood. It is mostly, everything is so new. I do hesitate to say something like this, but I would say it's probably mostly confined to the respiratory tract and respiratory tissue. Um, so yeah, so you concerned about
I,
treatment of
Yeah, well,
bodies
I'm
and how it could
personally
be
not
spread?
concerned, but I think that's another thing that I had come up is people are a little worried because of what's happening in some of these, some of these larger cities that are seeing heavy, heavy, heavy death tolls,
Oh, I
compared
see.
to
I
other
see.
ones. And then there's like other countries that are
Yeah.
creating pits, and stuff
Yeah.
like that. So,
Well, so I mean, COVID is an enveloped virus. So there are some viruses that are really protein cores, and I think those are probably more able to exist outside of a living human for an extended period of time. Any of the viruses that have that bud off of an infected cell that are coated in basically a cell membrane, they need to remain hydrated to stay infectious and really a somewhat limited temperature profile to remain infectious. Um, but I mean, like I get this question a lot, how long can virus X live on the surface Y? And I mean, there's no good answer to that because it depends on the humidity, it depends on temperature, it depends on the moisture content of the droplet. So I think it is, getting back to more dead bodies. I mean, just treat it is probably the safest bet is to treat any body as an infectious agent and treat it as if it were infectious.
yeah. And I think that's fair. I just, again, it's one of those things that a lot of people, there's a lot of bad information and a lot of people are scared. So,
Yeah.
right. So I think it's fair to address a little bit, especially if that information is out there. Now it
Yeah.
sounds like it's not as,
And it's not I would say the primary way the virus is spread. And the primary way it's spread is through, these kind of close contact, sneezes, droplets, things like that. So, the usual wash your hands, don't touch your face, is probably the main way you can protect yourself. Staying away from dead bodies is, I would say, lower on the list.
I think we already do that anyway. I don't
Yeah.
know how many dead bodies.
But if it's
we have.
unavoidable, it's unavoidable. Like you have to do an antibody test on a dead body. Well, then,
There you go. See,
just
now
take
when you go to
proper.
work tomorrow, you can have that conversation with the team and be like, all right, so
Okay, you
yesterday
gotta
on a
find.
podcast, I talked about testing antibodies on a
Yeah.
dead COVID body. Can we do that?
It's just a hypothetical in any situation. It doesn't have to be COVID.
So I think we'll shift gears a little bit, so the other article that you had sent over was about the vaccine, right? And
Yes.
so you're
Yes.
also participating in trials for a vaccine, correct?
Like I said, the shake and bake and I helped as far as I'm helping. Very,
Yeah.
very minimal helping, but I'm helping.
Yeah.
So in this particular trial, okay, so one thing I want to iterate is that vaccine progress is not fast and we don't want it to be fast. And even in emergency situations, we do not want this process to go quickly, and we do not want to cut corners. We don't want, I mean, this is not, I am a huge proponent of vaccination, but we don't want to move forward and inadvertently, let's say, trigger an autoimmune disorder in individuals, or let's say, as a purely hypothetical, we give someone a vaccine to this particular variant, COVID-19, and next year we get COVID-20, and they are more susceptible, more virulent, more aggressive, immune response, and things are worse if you got the vaccine. Like, we don't want to make things worse. So when we do vaccines studies, things are slow, things are we start in small animal models, we move into non-human primate models, we move into, I mean, you really call them phase 0 studies, where you just give a vaccine to a handful of individuals and you see, okay, nothing bad happens. And then you can slowly but surely start giving it to more individuals. And in this particular case, this study that's currently happening, so this trial is not an efficacy study. So we are not giving this vaccine to thousands and thousands of people and saying, hey, do the people who got the vaccine not get COVID disease as aggressively or as frequently as those who got the placebo? That's not what this study is. This study is still a phase 1. So what they're doing is giving the vaccine to healthy people, and again, looking for those adverse events and those serious adverse events that we mentioned before. And we're also going to be studying their immune response to see whether it looks like they could be making a response that could be protective. We are not challenging these individuals with virus, which we can do. So let's say we had, a norovirus vaccine. We could, in theory, challenge you with norovirus to see whether it was genuinely protective. That's not something we would do in this particular case. But we can look at the antibody response to see whether, in let's say we purify the antibodies from these individuals, and in a tissue culture dish, it can stop the virus. Cool. We can purify these things called cytotoxic T cells. So these cells will attack infected cells. We can purify those from these vaccinated individuals and see whether they can attack virally infected cells in a tissue culture dish. We can look for different cytokine messengers, and we can look for all kinds of signatures to see whether the immune response to this vaccine looks as if it would protect these individuals, but we are not looking to see whether these individuals are protected, not yet.
Yeah, because it's almost too early to try and detect that, correct? I mean, that would be crazy for us to run that fast.
And there's not enough people in this study to do that as well. So let's see, how many people are they ultimately enrolling?
They have 45 is the estimated enrollment.
So 15, 15, and 15 in each. So there's 3 arms to this study, and there's 15 individuals in each group. So when we do like a real study, there's going to be hundreds, if not thousands, of individuals enrolled in these studies. And so, like with the drug trial mentioned before, there are hundreds and hundreds of individuals in both of those groups. So this is still very, very preliminary. And also, let's say too, because there's only 15 people per group, we're not necessarily going to encompass all of the diversity of the human population in groups of 15. So that's another thing we need to take into account, is that the immune system of humans is incredibly diverse, and especially based on our geographic locations. So, we might have, in case of the HIV vaccine trials, they ran one in Thailand several years ago, showed minimal a little bit of efficacy. They tried to replicate that study in Africa, and they had to stop the study because there was no protective benefit whatsoever. So when it comes to the immune system, you can't just study 30-year-old white males. Like you need to make sure you look at men, you look at women, you look at all different kinds of ethnicities to make sure the vaccine is going to work for everyone.
Real quick question. Just so you mentioned the different ethnicities, and the
Yeah.
different genders and things like that, but
Yeah.
does age also play into that? Do you have to actually break it down and look at it? Let's just
Yes.
pick white women. We could say white women between the ages of 18 and 35 or something like that.
Yeah. So all of that is incredibly important when it comes to vaccine responses. So men and women will have very different responses to vaccines. And then your immune response changes a lot over time. So yes, all of that, yes.
Well, and I think, I just not trying to derail the thing or anything. I just think that that's important because
Yes.
that lends to understanding. And we've done a vaccine episode about 2 years ago with you, but it helped people understand why you can't just move this to trial, because there's just so many variables. Even if we didn't care about hurting people, there's so many variables to understanding how this will work. Can you imagine the mass hysteria if we said, hey, by the way, there is a vaccine and it affects 80% of the people will be covered by this. And then later on you find out, well, we didn't do enough studies and it really only covers 10% of the population. Not only
Yeah.
do we waste all that capital and everything else, just you create a larger mass panic than even needed.
There's also a lot of, the potential for all of those people who, thought they weren't going to be affected. Suddenly they're getting affected, and then it blows up in your face.
Yeah. Yes.
And then we have a
Well,
horror
and
movie
then-
on our hands.
And with vaccines, then you have people who turn around and say, well, that vaccine, I got that vaccine and that was bullshit, so I'm not getting another one. And then the next season's influenza it takes out everybody else who's left. So slow and steady. And nobody wants to hear slow and steady when, everybody's world has just been turned upside down. I don't know anybody who is business as usual right now.
No.
But and really, that's what we need. Like, we need a vaccine, and in absence of a vaccine, we need a drug, and hopefully they can find one off the shelf that works. But what we need are carefully designed studies, not. Listen, I understand the appeal of trying anything, but we need carefully designed studies to figure out what works and getting that to the people who need it.
Yeah. So you said they're breaking them up into 3 groups, and I think that's what this is right here. I'm assuming that's what the arms mean.
Yes.
Okay. Those are the 3 groups that you can see that they are breaking them up into. And is the only difference, or the only big difference, the fact that they're the amount of whatever they're injecting them with?
Yeah, the dosages, let's see, must be the micrograms of the mRNA. Yes.
Okay. And I guess I'll be honest, I don't know enough. I don't know enough about some of this stuff to know, is the mRNA, is this just like a generic name for this right now,
So,
or does it have scientific meaning?
usually, this is an extremely unique vaccine, and extremely unique in the sense that we don't have any vaccines right now that look like this vaccine. So normally when you get a vaccine, it is, let's say, just proteins that are proteins from a bacteria or proteins from a virus that are not infectious. They're not anything on their own. They're just proteins. And so you inject those proteins into you, and your body learns how to fight the virus or the bacteria by learning to fight those proteins, and you're fine. Alternatively, we have vaccines that are killed or weakened viruses and bacteria. So we tie the virus's hands behind its back and give it to you, and your immune system has no problem taking it down, and thereby learns how to fight the real virus. Okay, so those viruses are made in something else. They're made in other cells, and those proteins are made in other cells and purified and put into you. In this case, what we do is, so the mRNA, this is the message. This is a message that says, this is how I make this viral protein. And we put it inside of a fat. We hide the message in a fat and inject it into you. And that fat finds its way to one of your cells. And the message then is made by your cells. And so there are a lot of, I don't know how to say this. Your body has its own signatures as far as how it makes proteins. And that's how it would look if the virus really infected you. That's how that protein would really look. And so this is a way of getting your body to make that viral protein the way it would actually make it if you were infected, instead of making that protein somewhere else in let's say a tissue culture system in a laboratory and putting it into you, having your body make that protein. And so it's folded the way your body would fold it, and it's got sugars on it the way your body would put sugars on it. So it's very much tailored the way your body would make it. In theory, that would be good. But the question is, how efficient is this? What's your body's motivation to actually make a response to this? In theory, this could be really cool, but does it actually work? And how efficient is it? So that's part of what the study is going to answer. Because also in theory, you could inject this into someone and absolutely nothing would happen because your body doesn't care.
Yeah. Well, and I think that's important, right? Because that explain, I guess for me, that kind of says the difference between what, how your body absorbs this. And that's why there's a lot of people that get freaked out, there's a lot of people that get freaked out that, you're being injected with this weird stuff, and that's why, well, no, you're just getting injected with the dead virus in a protein.
Parts of stuff.
Yeah.
That your body will break down and get filtered out by your kidneys, and that's that. You hope you made. Actually, I think you mentioned a minute ago that before we started talking about the dead bodies, that you had to get a few booster shots of your vaccines. So that's the deal. That same thing happened to me. And I tell this story, I think every time I talk about vaccines, is that I had to get a third booster of my MMR because my body forgot mumps. Because I had antibodies to measles, I had antibodies to rubella. My body just forgot mumps because I just got the vaccine. I wasn't ever genuinely infected. And so that is part, or one of the drawbacks of vaccines. And one of the drawbacks of our immune response is that, sometimes our bodies just get lazy and forget things and need little reminders, little booster shots to perk, as you said, to top things off again. But and maybe in this case, the lowest dose, your body just completely ignores whatever occurs and is just unimpressed by the situation, whereas at the highest dose, it finally responds to the injection, and you get a nice, pretty antibody response. We'll see.
Neat.
And I think that's that's weird that you ever had to do that. Like, that's just weird. That's just weird. Like, I've never heard of that before.
No, it is somewhat common. Like the randomness of your immune system, and to this day, I still don't understand why creationists got so fixated on the immune system, because it is so wasteful. It is so lazy. It is just good enough. It is something a 12-year-old who stayed up all night playing video games, who didn't do his homework, would slap together at the last moment. It is not something an intelligent creator would do on purpose.
Well, that's I think the greatest argument against the idea of creationism is how imperfect our systems are. Sure, they work, and it's quite incredible when they work properly. But when you get down to it, I mean, like you saying your body forgot its immunity to mumps, my body forgot its immunity to rubella. So it's kind of random, but that also kind of explains, the importance of vaccines in terms of, herd immunity. So
Absolutely.
for those of us who, our immune systems got lazy, well, we're protected by everybody else's immune systems who didn't, and that magical 90 to 95% of the herd being immune means that we don't have to worry about our body forgetting, because everybody else is carrying that load for us because our body got lazy on us.
Absolutely.
So I'm assuming that this study has already been filled. I don't know, maybe not. But,
Yes.
Okay, so it's kind of funny. They talk about the eligibility. I think we kind of touched on that already. I mean, I don't know. I don't really know how much we have to worry about that. They do say they have some exclusion stuff, and there is quite a few things in here, like female women that are breastfeeding.
Yep.
And is that just because they're worried about it transitioning to them?
I have no idea, but it's one of the usual ones. If you're pregnant, trying to become pregnant, we don't want to mess with any of that. I think that's probably a usual disclaimer or usually studies.
Okay. And that's fine. I mean, it's one of those things where you're just like, why? So they, the other ones is like, you can't talk to medical, if you're under psychiatric treatment. I don't know. Is that a common one too?
I'm not sure about that one.
Okay. Because that one seemed a little weird to me as well. But I guess, go ahead.
Looking into the one that you just mentioned there, presence of self-reported or medically documented significant medical or psychiatric conditions. In one of them, one of the requirements or exclusion things, whatever, they're talking about just respiratory diseases. So I think that they just don't want to have anything that can possibly confabulate the results on that one. So I think that would just be kind
of a panic. Like, let's say you have a panic disorder, perhaps, and you concerned you get this, or you might have like you even get concerned that you might have an anaphylactic response to the vaccine or something like that. You might have a panic response to that. Or I don't know. This is me just making things
or
up.
drug reactions, that type of thing. Because like I know there are certain antibiotics that can nullify hormonal birth control. So they just my assumption here is that they're just trying to reduce the amount of data that can can warp or twist whatever, because this is being such a low number of participants. It's really just, it's not about the grand scheme. It's kind of just making sure that it works for specific more ideal conditions would be my assumption.
A surface level pass.
Yeah. You got better science terms than I do.
Oh no, we're just making stuff up.
I think that's how that's supposed to be. I think if
But
we
it
had better
sounds better
than her, we'd have problems, right?
It sounds better though. Oh, I would actually be excluded from this. Neat.
Would you?
I mean, or at least there's a good chance they would be. Who told you? Stop reading my diary. No, is because I have an underperforming thyroid. And I'm on medication for it. Yeah. Neat.
So anyway, I don't know. It is neat. That's fine. It's good to know you can never do trials, right?
But not
anyway,
we would want you included in the big ones.
Yeah, I like that. I can do that.
We want the diversity of everyone, because everybody's going to need to get it. So
this is true.
So I guess as we.
Oh dear.
Oh, and I think we've covered both of these, right? I don't think we have another article. You didn't have another one?
No, those were the two main ones that I wanted to make sure you all got to see.
Yeah.
just so you can see what what does a drug clinical trial look like? What does a vaccine clinical trial looks like? I wanted your viewers to be able to click on those links and peruse them themselves.
Yeah, and I feel like I said that was the whole point of this extra episode was specifically that, right? It was specifically to go through to have the conversation, about what's going on right now
Yeah.
and what we can do, to get better information into the public.
Yeah.
So we won't do quick saves because Hannah's not here. So, and I'll be honest, I don't have another quick save anyway, especially after my long quick save from last week. So, but Abby, it was a pleasure to have you on. Thank you so much for joining us. And you're welcome to come on and update anybody that you would like to over the course of the next couple of months as this thing progresses, right?
Oh, I hope it progresses that soon.
It was
Have a
wonderful
night.
to meet you.
Oh yes, it was nice to meet you too, buddy.
Yeah. And I think I agree with you. I hope it progresses a lot faster, than we, imagined, and that people stop dying and that curve gets flattened a lot quicker. Especially for those of us who don't get a choice and have to go, to work or have to leave the house.
Yeah.
My company still, all our warehouses are still running because they're all we all house essential material. But I don't know. For me, it's been a journey. So, do you have anything you want to share real quickly before we end the show, Abby?
Everybody, just keep washing your hands. Don't touch your face. Stay home if you can. And yeah. And just know that, we're, like I said, we're all feeling it right now. Everyone's lives have been completely disrupted, and I feel for you, and we're trying.
Awesome. Thank you all so much for joining us tonight. Again, it was a pleasure to have somebody with such knowledge on that can actually talk to us about these things, since we can't seem to get the current administration to join us and have true educational information at all. So thank you all. Again, this has been a special Cellar Door Skeptics podcast. We don't normally do midday ones, but since I work from home and Buddy was able to join me today, and Abby works very early in the morning, so we were able to do a weirdly lighted show where there was absolutely no drinking, at least on my behalf. So that was like a first.
Not on mine. Weird. Who are we? I don't know us anymore.
I know, it's kind of weird, isn't it? We typically, usually by now, the hour into the show, we've had a couple of pops and yeah, but today, not so much. So anyway, thank you all so much for joining us. The links are in the description of the episode. You're able to find those at all of your favorite locations. Make sure you give us a rating on iTunes. We don't ask for Patreon subscribers right now because we know people need to be saving money, but we would appreciate if you went and gave us a 5-star rating or any star rating on iTunes or Spreaker. Thank you so much, and have an amazing night, everyone.